Hi, I'm Dr. Brian Kaufman, a retired family doctor and a CLL patient myself, and the co-founder, executive vice president, and chief medical officer at the nonprofit CLL Society. And I'm here in New Orleans at ASH 2022.
I'm James Blachly. I'm a physician scientist at Ohio State. I study the genomic changes that lead to leukemia, and I'm very interested in both the changes that people are born with as well as the ones that they acquire that might lead to diseases like CLL.
So I love interviewing physician scientists because you're the ones who help us unpack the biology, and once we crack the biology, then we can figure out how to target it and improve the disease. And this is exciting because this is a drug that's so new that it doesn't even have a name yet. It's still letters. So I'm going to read the title of this, and then I'm going to ask you to explain it in sort of patient-friendly terms. And this is an oral presentation, which is difficult to get at ASH. The most important papers are reserved for an oral presentation. So obviously your peers were excited about this paper. Its initial results from a phase 1/2, so very early study, dose escalation and expansion study evaluating MS-553, a novel and selective PKCβ inhibitor in patients with CLL, SLL. So you're going to have to explain a little bit about that to us, because our patients are sophisticated. They know about Bruton tyrosine kinase, they know a little about BCL2 and venetoclax, but I don't think they know much about PKCβ inhibition. So set this up for us. Tell us why you decided to do this study, what drove you to look at this particular molecule, and then we'll talk about the study itself.
Yeah, it's a great question. This is a part of a pathway that everyone is already familiar with. So going back a few years, let's look back to 2012, let's say, when ibrutinib was being developed in CLL. We know that the B-cell receptor pathway is critical. The signaling through the BCR pathway is critical in the pathogenesis of CLL. And the signal begins at the cell surface, and the B-cell receptor signals through BTK, or Bruton tyrosine kinase, of which we have inhibitors like ibrutinib, acalabrutinib, etc. Next, through a protein called PKCγ2, which we may have heard of, and I'll talk more about that in a moment. And finally, through protein kinase C beta, or PKCβ, which is the target of MS-553. Now, around this time, we were beginning to see patients treated with ibrutinib on clinical trials at Ohio State who were becoming resistant to ibrutinib. And as you may know, we were the first to discover BTK and PKCγ2 mutations driving resistance to ibrutinib. We hypothesized that as signaling passed through BTK, PKCγ2, and PKCβ, that inhibition of PKCβ might be a way to overcome resistance to not only ibrutinib, but whatever might come next.
Well, that's incredibly exciting, and I'm going to just share personally, I was one of those patients, and I remember Dr. Bird, who was taking care of me in a phase 1 clinical trial of ibrutinib, how excited when these assays became available, looking for mutations where the Bruton tyrosine kinase inhibitors would bind, but also downstream gain of function mutations that would turn the pathway on again after it had been turned off, PKCγ2, which, by the way, I have. So I have a very personal interest in the story that you're going to tell. So now I'm understanding it's all part of that BCR, B-cell receptor pathway, but it's closer to the nucleus, a little bit further from the cell wall, and therefore some of the problems that might stop a BTK inhibitor from working, this may still work in those circumstances. Am I getting that right?
Yeah, that's right. And we and other people also showed that PKCβ is critical to drive the pathogenesis of CLL. So not only does it signal through protein kinase C beta, but PKCβ is upregulated. And so work done at Ohio State and MD Anderson and other places in the laboratory showed that this protein was essential. And not only that, but there were earlier PKCβ inhibitors tested both in the laboratory and in the clinic that showed that inhibiting PKCβ could antagonize CLL. Unfortunately, the development of PKCβ inhibitors has been difficult because of toxicities. And this is because PKCβ is expressed widely in essentially all of our cells. There are different isoforms. And
remind us what an isoform is.
There are different forms of the protein which are almost identical, but in different cells there might be a different isoform. In this case, we know that PKCβ is important in CLL cells, where something like PKC delta could be important in T cells.
So tell us a little bit about the study design. What were the patients that you were studying?
Yeah, so the study design is using this molecule MS-553, which is a relatively selective PKCβ inhibitor in patients with not only relapsed refractory CLL. We were looking for relapsed refractory patients who were high risk and had mutations in BTK, PLC gamma 2, and even TP53. In fact, we allowed patients who were not previously treated but who had a P53 mutation to enroll. And so it's a very heavily pretreated patient population who is targeted and is enrolled in the monotherapy cohort of this study.
Well, you piqued my interest when you said that there was toxicity issues, so I'm sure when all phase 1 trials, that's the first thing you're looking for. So what did you find? Did you find this was a molecule that people tolerate fairly well?
This is far more tolerable than prior PKCβ inhibitors like sotrastorin, which is one that you might see in the literature. And the primary toxicities that we observed in this study were gastrointestinal in nature, with no more than grade 3 toxicities.
Remind us what grade 3 is.
Yeah, the grading system tells us just how severe a toxicity is. And by the time something gets to be grade 4, it's considered pretty serious and can halt development of molecules.
So most of these were more mild, and this would be things like stomach upset, diarrhea, that kind of thing. Is that correct?
That's correct. So there was a lot of it, but it was quite mild.
So the most important question from a patient's perspective, did it work? What kind of results? I know this is very early in the trials, aren't designed to look at efficacy, but we also know that we always look at efficacy at every stage of a trial.
Great, great question. We were able to evaluate efficacy not in the entire 43 patients who were enrolled and included for safety data, but in a subset of 23 patients. And given how heavily pretreated these patients were, how sick they were, how many prior therapies they had, there was a median number of 4 prior therapies, which with up to 12 prior therapies. So that's a lot of treatment.
That's amazing.
It's a lot of treatment. But we still saw a combination partial response and partial response with lymphocytosis rate.
Remind us lymphocytosis.
So yeah, in the BCR pathway, when we inhibit the BCR pathway, whether with a BTK inhibitor or a PKCβ inhibitor, we see a release of white blood cells, CLL cells, into the circulation from lymph nodes and wherever else they may be hiding.
So that causes the lymphocyte count to go up, and that's called lymphocytosis. Yeah.
Correct, correct. So for response purposes, we recognize that that doesn't represent disease progression, and we combine PR, or partial response, and PR with lymphocytosis. And in this study, we saw a 48% PR/PRL response rate in this very heavily pretreated population.
So almost half the patients were responding in this very difficult to treat population. Are there any is this trial still open? Are you looking to expand or move further?
It is still ongoing, and in fact, we have finished the dose escalation portion and we're expanding. And not only do we have a monotherapy arm, but we also now have combination arms. We have combinations with acalabrutinib, including in the frontline previously untreated setting. And we have a combination of MS-553 with venetoclax and a CD20 antibody, also in the first line or second line setting.
Wow. So these would be on clinicaltrials.gov. We can give a link to the
That's correct. Patients. That's correct.
So this is very exciting, and I'm very interested. Just any final thoughts or conclusions you'd want to share with the CLL patients?
Yeah, I think this represents a great way of thinking about drug development, and that is to anticipate mechanisms of resistance, potentially years before they're reported, and work on finding a way to target them. One of the other things that we'll report is that this drug may be effective even against mutations induced by newer reversible BTK inhibitors like pirtobrutinib. And by inhibiting downstream of the non-C481S BTK mutations that we can see after treatment with pirtobrutinib, this drug can still be effective.
Well, I'm very excited about this, and I started by saying, you know, I love interviewing physician scientists because you get it, you have a patient in front of you, you know how to take care of a patient, you know the art of medicine, but you also know the science of medicine, and without these drug developments, we'd still be treating CLL with chemoimmunotherapy and cytotoxic drugs that are non-specific. I'm so grateful for what you're doing.
Thank you so much for inviting me today.
Thanks.